I think an insatiable curiosity is a quality that's common to every scientist, especially the ones who are successful.
If you're trying to look for interesting projects, interesting questions, the more fundamental, the better. The more interesting it is, the broader the implications.
Dr. Szostak's work is on the development of artificial cells. So he's working at the cusp of chemical and biological evolution. So it's really neat to talk to somebody like that. Jack Szostak is a biologist. His prize-winning work was on telomeres, which are caps on DNA that explained a lot of things about how the translation of DNA works. His current research interests are in the origins of life.
Once life gets started, it can adapt and evolve and colonize many, many different environments. Our astronomer friends discovered already over 400 Earth-like planets. And the question of whether there might be life out there in those other worlds really boils down to the question of whether it's easy or hard to make that transition from the chemistry of an early planet to the beginnings of biology.
Your research, but from hearing you speak yesterday, is very much curiosity-driven.
Right.
And no real practical application.
I
it's
think
just
so.
like
Not for a long time anyway.
Right.
Nothing I can foresee at the moment.
So as a young scientist, it seems like there's a big shift away from that. Most of the funding, most of research it's industrially driven or it's applications based, and it seems like a really fundamental change in the way we think about science and the way a scientist's career is going to be shaped. Do you think that change is as drastic?
It's a little hard to find the balance, and it shifts back and forth. I think right now there's maybe a little too much emphasis on the translational work for my preference. For example, in the work I do that led to the prize, it was very basic research. At the time, we were just curious about some DNA mechanisms, had no idea about importance in biology or medicine. And, over the years, these things just sort of emerge.
Right.
If that kind of basic research isn't supported, then eventually the pipeline will run dry.
So, pertaining to the origins of biomolecular homochirality, this is a pet peeve project.
We were talking about the issue of the kinds of molecules that came together to make the first living cells, and in particular, the fact that a lot of these molecules come in two forms that are mirror images of each other. But in modern biology, we generally see only one of these mirror image forms. So there's been a long-standing question as to how that got started. How on the early Earth did we end up with having only one of the two possible mirror images for the fundamental building blocks of the first cells.
Chirality comes from the Greek word for handedness, and that actually provides us with a ready analogy because to be non-superimposable on your mirror image, you can see that your hands are mirror images of one another. But when you superimpose them, your thumbs stick out on either side. So they're non-superimposable on their mirror image. And molecules at a microscopic scale have this same property in 3 dimensions. Biomolecular homochirality is a fancy term for a pretty simple idea. So amino acids and sugars, which are the building blocks of life, are all, almost completely, when they're found on terrestrial Earth, one hand. And there's not any real explanation for why this is so.
20 years ago, it just looked so impossible because there were almost no mechanisms that were well known. And now we have so many different ways in which the problem could be solved that it's almost embarrassing. So since you've worked so much on chiral catalysts, have you thought about ways in which there could be chiral autocatalysis in prebiotic chemistry?
So the examples that are out there really don't explain it, that's for sure. So, I mean, a dialkyl zinc reaction is not going to happen under prebiotic conditions. So that's not.
But it seems
really
worth trying to think
applicable.
of reactions that could be.
There's some other interesting things then that I guess are way outside both our fields about, you know, what established that chiral bias in the first place. So there's some really interesting stuff about using just physical forces to impose chirality, and so.
Oh, you mean like the magnetic?
Yeah, or
effect?
circularly polarized light is
right.
so.
Well, that's the usual explanation
Right.
for the extraterrestrial stuff, the meteorite stuff, right?
The Murchison meteorite provides significant evidence that the origins of biomolecular homochirality could be extraterrestrial. It landed in Australia in '69, and it had a number of what we would call non-terrestrial amino acids. They were very, very similar in structure, but just had one small structural change from the building blocks that compose our bodies and, you know, everything else, that all other living things. And they exhibited a small enantiomeric excess. So instead of a 50-50 mix of right and left hands, they had a small excess of one over the other. And that excess favored the chiral sense that we see in the terrestrial amino acids that compose living things on Earth. So the amino acids that were on the Murchison meteorite that exhibited this small enantiomeric excess, they were all alpha methyl amino acids, and all of the terrestrial
that's what
amino
lasts.
Exactly, right. So if that was the source of our initial chiral bias, that means that there would not only have to be a mechanism for amplification, but first the mechanism for a reaction of that non-terrestrial amino acid into the ones that we see in our bodies. Otherwise, you know, we would have alpha methyl amino acids in living organisms.
I'm not sure I buy that.
No?
I mean, you could have something that makes normal amino acids chiral. Of course, that wouldn't last for 4 billion years, but it doesn't have to, right? It just has to last long enough to come and have an influence in the right environment on the early Earth.
Well, yeah, it would have had to have happened on Earth. I mean, do you really think something could make it through the atmosphere as a racemizable stereocenter with a?
Well, when the Murchison meteorite fell to the ground, if I remember the stories right, it was still cold in the middle, right?
Really?
I mean, you got this skin formed around the outside of it.
So something that's conserved in the.
Yeah.
A few of his opinions surprised me. There's some theories that the chiral bias of amino acids came from some extraterrestrial origin. And the nature in which they were delivered, so the molecular form, I've always thought could only be a non-racemizable alpha-methyl amino acid, which is just a bunch of jargon. But he actually had some other opinions about that, that the temperature of the inside of a meteorite delivering these type of molecules would stay cold enough that that wouldn't be a problem. Your prize-winning work on telomeres, that work is a phenomenon that applies to all different forms of life. I just wonder if that sort of, I don't even know if this is a word, but fundamentality kind of draws you in your work? Is that something that you feel like?
Absolutely.
Okay.
I mean, I think if you're trying to look for interesting projects, interesting questions, the more fundamental, the better, right? The more interesting it is, the broader the implications.
Okay.
And so thinking about the origin of life fits perfectly into that.
Sure.
It's not much that's more fundamental.
Right. I like thinking of our work in a bigger perspective. I mean, when I say our work, I mean like just very generally as like, you know, the scientific community.
Right.
And it's a very palatable idea to have you know, the kind of like aesthetically pleasing things that we see in culture and the things around us, see those same things manifested in science. And
so right, it's one of the things that drives me crazy is when people criticize science for taking away this perspective of natural beauty. But, you know, just as we look around and everything we see here is so beautiful, when you look at the structures of molecules, as you can appreciate, they're very beautiful. So I think if you know more about science, you can appreciate beauty in different places.
Right.
And there's always surprises. You
Yeah.
always see new things, and they're wonderful.
Yep.
So.
Yeah, I think of science as the substance of aesthetics.
Exactly.
It's the
Yeah, I like that.
details.
I
Yeah,
like that.
so,
Right.
yeah. That was a fantastic experience. It was, I don't know, I'm a little bit speechless. I'm very excited. I already feel like I have a fresh perspective. Day-to-day bench work gets really tedious, and I just feel really excited about what I do again, and not that I ever really lost it, but you know, a renewed vigor.
Even though we're doing very different kinds of experiments in our labs, because our interests do have this area of overlap, we had a great conversation. It was very fun talking to Abigail about all of these ideas. She's thought broadly about fundamental things like the origin of life.